Hello, everyone. Thank you for joining today's webinar, Global Perspectives in Metabolomics. I'm Zhao Yan, and I'm truly glad to connect with colleagues from worldwide. I work at BGl Genomics as a field application scientist, where we collaborate closely with academic and clinical partners to translate multi-omics data into biological insights. In this session, I'll walk you through ways the place of metabolomics within multi-omics precision medicine, analytical strategies, untargeted, targeted, and semi-targeted, workflow and data quality assurance, integration across omics layers, and a few case studies and closing remarks. So let's begin with the big picture. As we all know, the era of precision medicine depends on our ability to capture individual biological complexity. We are aiming to analysis propels precision medicine by enabling personalized and accurate disease classification, treatment selection, and clinical decision-making. Researchers are trying to integrate as more data as possible for genome, transcriptome, proteome, and metabolome from healthy and disease and pre-disease or disease group of people, from which data to the further make precision prevention, diagnosis, treatment, and prognosis realistic. For example, a study with multi-omics data built a biological function model by data analysis and successfully trained the data to distinguish certain type of disease development. At the same time, giving supplement to some of patient, rematching of the personal biological function model was found. With this integration, it provide us with helpful information of theoretical foundation for improving our understanding of health and disease. But while genomics and transcriptomics are now routine, downstream omics like proteomics and metabolomics remain more challenging. Why? Because they rely heavily on mass spectrometry, producing large, complex datasets that require sophisticated interpretation. On one hand, metabolomics, and proteomics are more complex compared to genomic and transcriptomics, with a wider variety of basic structure, with a wider variety of basic structures and the actual type of metabolites and proteins. On the other hand, the mass spectrometry instrument used for these studies generate much larger data and more complex data sets, heavily relying on database for interpreting and analysis. For example, in a typical mass spectrometry iron chromatogram, each peak presents one or more similar substance, and only by refreshing the specific instrument's database can further analysis be performed. The entire process, from wet lab to data analysis, needs to be fully integrated. This complexity often causes confusion and difficulty for many researchers. By optimizing both experiment design and workflow, we can make metabolomics approachable, reproducible, and deeply informative. That's exactly what we focus on at BGI. We need a systemic experimental setup and optimized workflow for that. In following, I will begin with wet lab for data generation, dry lab for data analysis, and two deep dive case study of multi-omics. The first step for generating the data is the appropriate instrument and method selection. This is based on the experimental purpose and sample type. I'm taking an example of our BGI lab. We have more than 45 different kinds of instruments, but it's not some collecting game. The truth is, each instrument or specific combination of any of them has specific limitation and strength regarding accuracy, dynamic range, resolution, and the speed of analysis. Here you can see Here you can see our main MS instruments: thermal orbitrap, Astral, and Lumos, and Bruker's timsTOF Pro, and SciX QTrap, or integrated with DNBSEQ sequencing for multi-omic projects. Having complementary MS technologies allow us to tailor each project, whether it's lipidomic, metabolomic, or proteomic, to its biological question. We are now taking advantage to deep screening with high-resolution MS to untargeted screening, for example, with QE. But considering the speed of analysis, we will use QTrap, the triple quant MS for target panels. Anyway, there will be one most appropriate option for your own demand. Let's discuss the strategies: untargeted, semi-targeted, and targeted. Untargeted metabolomics, an unbiased survey capturing thousands of metabolites, ideal for discovering unexpected biochemical shifts. Its strength is broad coverage. Its challenge lies in complex data processing. Proper LC column selection and draw iron mode scanning are key for success. Then the targeted metabolomics, hypothesis driving, and quantitative, we focus on specific metabolite groups, for example, amino acid or bioacid, quantified using isotope labeled standard. High producibility and sensitivity make this approach perfect for clinical validation, and the semi-targeted metabolomics bridge the two. Using procedural target MS method, we achieve wide coverage with improved quantitation, ideal for large-scale cohort studied. So discovery, quantification, and throughput all balanced in one box. Untargeted metabolomics is the most exploratory method, ideal for discovering unexpected metabolite changes. It provides unbiased coverage of thousands of metabolites. However, its complexity lies in data processing. The wide dynamic range of metabolite and variable aerosolization efficiency make quantification analysis difficult. There is no signal analytical tool of detection technique can cover all of them. For this reason, choosing the right LC column is crucial. The stationary phase chemistry should match the nature of your ideal analysis. We have concluded 4 different types of column for different physical chemical compounds. For example, the C18 is more suitable for nonpolar and medium polar and medium polar small molecules like hormones, fat soluble vitamins. But for strong polar molecules like amino acid, you will need to use an amide column. For plants or lipid, we also have other columns with specified anti-capping for the stationary phase. Then, after the LC phase, the compounds will be performed with fast scanning mass analyzer operating in the wide mass range and need a powerful data bank for compound matching. A process of data analysis is showing here. For identification stage, the spectra of each compound should match with the database and score how fit they are. In BGl, we have our self-built in-house library and local database, totally with 10,000 compounds information saved. This databank is strictly aligning with their method, including gradient time, solution used, the LC column, or the energy of ESI. Compared with other databases, the identified rate can reach 60% of detection. After identifying Data should be normalized and well prepared for differential expression analysis. At the end, we can put the differential expressed metabolites back to the pathway map and discover the change between treatment or disease groups. For metabolic studies, there are some public databases like Chemic Spider, but these databases only have very limited information for specific metabolite, which is the MS1 spectral information. So actually, this information is not enough to do 100% confirm the identification result. In order to solve this issue, we also built our in-house standards database for plant and animal and human and microbiota. In the standards database, we have these three aspect information of metabolites: the retention time, the MS1 and the MS2 spectra, and so combining the usage of these three information, we can 100% confirm that the metabolite detected is the metabolite in the database. So this means that these identification results are with very high reliability. Going back to the other type of analysis, the targeted approaches. It starts with an assumption that selection a group of compounds and targets a specific aspect of biological system. When you already know which pathway you are interested in, targeted metabolic is the right choice. For example, the energy metabolism pathway or all the neurotransmitters. It in general covers less than 50 compounds. The semi-targeted metabolomics is with several internal standards and can calculate the concentration like normal targeted, but it is more widely set up for screening like untargeted. The HM Pro panel at BGI is an representative. It covers 1,000 of metabolite and 700 lipids. Researchers can get all these identified and qualified at once. Okay, I give a short summary of the untargeted and targeted panels we have. You are welcome to send your list of wish to us and we can combine for you. The second part, we move to dry lab, the data analysis. In MS-based omics data, raw quantitative data must be pre-processed to enhance quality before statistical analysis, ensuring more accurate and reliable outcomes. For targeted or semi-targeted analysis, a normalization step for calibration curve is needed and then calculate the concentration. In these two approach, the data reprocess is still not complex, but the untargeted approach, we will need to confirm the instrumental QC, sample QC, and data normalization and retention modification, several steps before we can use the batch data to be compared. For example, it would include overlaying of the eternal standard PPC, overlapping PCA score plot and heat map of QC samples. You may not familiar with this map, which is exactly what I am coming to answer. How could we work with this huge amount and complex data? Data visualization can be integrated at many stages of the data processing and analysis workflow. It may be used for exploratory purpose to gain an understanding of the data's underlying structure and patterns. It may help visually compare samples and identify outliers. For example, PCA is a method of obtaining a few comprehensive variable. So to say, reducing of dimensionality, we can use for plenary observation of group to or between group differences. PCA is unsupervised means we are not considering grouping information in this stage, which can help us to find abnormal samples. Then the PLSDA is another similar method, only it is supervised. We use the grouping information to build the model and manually classifying samples. The classifying is better than PCA and can be used to screen different metabolites. For judgment of the metal of the model, it will also calculate the validation plot. And for differential expression, the volcano plot is an effective visualization tool for differential expression research because it combines statistical significance with amount of change, making it simple to find metabolites with significant differences between experimental conditions, like we are showing in the color green on the left side, and as the downregulated metabolites, and the other side, the red colorized, the upregulated. The gray point of metabolites means no significant difference, and the more far away of the point, the more significant expressed in the group. Heat map provides another visual representation of multi-dimensional data, displaying variables in a matrix format with colors including relative value according to a scale. This method effectively reveals similarity and differences across multiple variables, such as the relative abundance or intensity of feature in MS data, making it a useful tool for highlighting upregulated or downregulated patterns across samples. Mainly with these two set of map, we can distinguish those differences between groups. Then we will reference the database like functional or pathway. Bubble diagram can show the significantly changed metabolic pathway. And with the pathway map, you can find the location and relationship of them. You can see also relative metabolites and enzymes and so on. Talking about so much tools, if it's still too much script for our researchers to run through the data analysis, BGI provides a web-based solution for the convenient analysis, visualization, and interpretation of data. Your omics data from RNA, protein, and metabolites can upload to the platform and finish all the analysis I just mentioned before. In each map, like the volcano map, you can click to get the information of the metabolites you are visiting. Only a few clicks without any script to be written, you can get a rough idea of your data set in few minutes. In total, you can simply send your sample and specify your analysis needs to us. Then we will take over the rest, and you can be more focused on your own research. Now let's take a step further beyond metabolomics alone. For the next section, it will be great to share some case study to let you know more how to apply this to some research. Single omics can only provide an incomplete picture of the object we study, while multi-omics can help us understand its full picture. For example, RNA and proteome is one of the most famous combinations of omics. We know that there are several steps between DNA expression to protein synthesis, and we even know that what is the code for mRNA to amino acid, but in many cases, they are not always expressing together. It may be measurement noise interference or false positive, or it would be more complex mechanism in biological models. Researchers can use to examine the multi-level regulation of gene expression and obtaining a global respective of gene expression. The correlation analysis panel we provide is also in this purpose to discover a certain mechanism, pathway, and elucidate a process. The difference expressed gene and proteins will be analyzed separately. The correlation between them will be demonstrated to different method and also the functional enrichment. The network, there are several common integration models. Protein and transcriptome like links enzymatic regulation to metabolic fluxes. Transcriptome and metabolome uncovers transcriptional control of metabolites, and metagenomic and metabolome connects gut microbiota activity with host metabolism, and the MGVAS means metabolite genomic-wide association study. There is a common workflow of metabolomics and transcriptome correlation analysis. Firstly, we will run these two omics separately and then do standard analysis. For transcriptome, we will get the information of differential expressed genes, and for metabolomics, we will get the information of different metabolites, and then we will do the correlative analysis using model like univariate correlation or unsupervised model or supervised model, and further biological function correlation. There is a research case study on mitochondrial energy metabolism on hypertrophic cardiomyopathy. Mitochondrial sample were obtained from both HCM patient and control group and were subjected to transcriptomic, metabolomics, and lipidomic profiling. Integrated network analysis revealed dysregulated myocardial fatty acid metabolism and isocarnitine metabolism in HCM patient. Additionally, the overall energy expenditure in the myocardium of HCM patient was increased, characterized by reduced level of high energy phosphate metabolites, along with decreased expression of mitochondrial related genes involved in creatine kinase and ATP systems. Another deep dive case study I'm showing is the relationship between intestinal flora and derived metabolites and human disease, means metagenomics and metabolomics correlation analysis. It was found in many kinds of disease, both intestinal flora and blood metabolites profile change. Metabolite generated from intestinal flora can be absorbed by intestinal epithelial cells and enter the bloodstream, and then they are directly or indirectly associated to health system or disease development. To simplify this research idea, the discovery and verification is the most preferred in these publications. The separated analysis of metagenomics and 16 sedimentation or metagenomics and find the relationship. Another validation set of animal experiments can be done. In one way, the facial microbiota or single microbiota transplantation or metabolite supplement or inhibitory, and see if any changes in the disease development. An example of this is a research on type 2 diabetes. The medicine berberine and probiotics have inhibit or sub-influence to specific type of flora, and they have found some short-chain fatty acid in blood also changing together with the flora composition, which showed how microbial metabolites, especially secondary bile acid and short-chain fatty acid, influence host physiology and disease. These illustrate how integrated omics can move us from observation to mechanism, from what changes to why it changes. Okay, let's talk about BGl Group. BGl Genomics is the eternal part of BGl Group, founded in 1999 as a research organization to support the human genome project. Over the years, BGI's research has contributed to the development of the field of genomics in significant ways, as evidenced by a wealth of peer-reviewed publication in prestigious scientific journals. Today, BGI is actively participating in the field of cancer research, biosustainability, and personalized medicine through a variety of subsidiaries and a global network of collaborators. We have more than 6,000 employees offer service in more than 100 countries and regions. As I showed before, BGI offers comprehensive multi-omics solution covering genomics, transcriptomics, epigenomics, proteomics, metabolomics, and integrated multi-omics analysis. Our service utilized high-throughput sequencing platform such as DNB-Seq-T7 and DNB-Seq-G400, and mass spectrometry technologies, including Orbitrap and QTrap, supporting applications from whole genomic sequencing and targeted metabolomics to across-omic correlation analysis. And besides this cutting-edge instrument and experience, we also have a scientific experience team. Our mass spec team was founded by Professor Liu Siqi, who is also the co-founder of BGI Group. Our platform was built over 20 years ago, almost together with the sequencing platform. Today, we have a lot of employees with PhD and master degree. With our specific experience in mass spectrometry, we handled over 10,000 projects per year and have more than 300 publications. Here are some representative papers. Quality control is at the heart of producible science. In every project, we apply 4 major stages of QC. It starts from the sample status check. For example, if you send the plasma or serum samples to us, we will firstly check if there is hemolysis, because hemolysis is not recommended for the metabolic or proteomic analysis. Then during the sample preparation, we will prepare both the QC and QA samples. The QC sample is to ensure that the instruments are running stably. The QA sample are prepared to using the sample of our own lab, and they are mainly dedicated to ensure that the technical operation are correct during the workflow. And then after we prepared these QC and QA samples, we will run them together with the real samples. And then finally, in the data analysis, we will first analyze the QC sample. Like I said before, the overlap analysis and PCA-CV heat map to check the QC sample's performance. So only after the quality control passed, we will move forward to analyze the customer's samples. Let's look at an example of large-scale project. In one human plasma study involving 2,000 samples, 2,000 samples and 350 QC over 70 years of MS acquisition. Our quality metrics remained extremely stable. Internal standard RSD is less than 10%. QC sample overlap was nearly perfect, and the consistent PCA clustering across 9 batches. Minimum QC correlation coefficient of 0.975. At BGI, our goal is to empower the scientific community with accessible, high-quality omics service, so you can focus on the biological that matters most. Whether your goal is biomarker discovery, disease mechanism exploration, or personalized health assessment, BGI is ready to support your journey. Thank you all for joining today's session. If you have any question or would like to discuss your specific project needs, please feel free to reach out. You can send us an email or visit our website. Thank you again for your attention, and I wish you all success in your future research.